Hey everyone, my name is Maria Lopez, and today I'll be talking about coffee consumption and the gut microbiome, a global connection. So let's get started. So the article I chose was published in Nature Microbiology in December of 2024. So it's a really new article. It's titled Coffee Consumption is Associated with Intestinal Lawsonibacter, Asaccharolyticus, Abundance and Prevalence Across Multiple Cohorts. And as the title implies, they are focusing on the relationship between coffee consumption and the human gut microbiome, specifically this bacterium Lawsonibacter asaccharolyticus. On the slide, I have the citation, and at the bottom, I have a direct link to Nature Microbiology. So let's get started by addressing the bluff, the bottom line up front. So, why should we care? Well, the gut microbiome plays a crucial role in health and disease. And we know that diet is one of the biggest factors shaping its composition. While many studies have explored the impact of foods like fiber and fats and sugar, less is known about how specific beverages like coffee influence the gut bacteria. And I would say the core research question is, how does coffee consumption influence the composition and the metabolic activity of the human gut microbiome, specifically in regards of Lawsonibacter asaccharolyticus? And the key findings, they were able to discover that coffee drinkers have significantly higher levels of Lawsonibacter asaccharolyticus in their gut, suggesting that coffee may actively promote the growth of this bacterium. And they were able to achieve this goal. They were able to demonstrate a strong, reproducible association between coffee consumption and the abundance of Lawsonibacter asaccharolyticus across large cohorts. And I think they were able to contribute to the science world by identifying Lawsonibacter asaccharolyticus as a key coffee associated gut microbe. For some background, I wanted to talk about coffee. So it's approximated that 2 billion cups of coffee are consumed worldwide. And coffee may have health benefits due to its complex bioactive composition. And a bioactive compound is simply a type of chemical substance that exerts an effect on a living organism. So, coffee has over 1,000 biologically active compounds. I listed a few on the slide. Caffeine, it's a potent stimulant. It affects the central nervous system. It's what gives us the alertness after drinking our cup of coffee in the mornings. Chlorogenic acid is a powerful antioxidant with anti inflammatory effects. Quinic acid modulates antioxidant metabolism and it supports DNA repair. And many others, a few others are polyphenols, nicotinamide. They all also contribute to coffee's diverse health effects. And so some studies suggest there are protective effects against type 2 diabetes, cardiovascular diseases, and even certain cancers. So coffee's role is not just as a dietary staple, but also should be seen as a modulator of health through the microbiome interaction. And so I also wanted to talk a little bit more about Lawsonibacter asaccharolyticus. So it was first identified from a fecal sample of a healthy 41-year-old Japanese woman. So, in this 2018 study led by Sakamoto, they took those scanning electron micrographs of the cell that I have on the slide. Um, they found this bacterium to be obligately anaerobic, gram positive, non spore forming, straight rod shaped organism. And they used 16S rRNA gene sequencing analysis, and it showed that it was a new genus and species within the family Rumococaceae. And so the most important and most fascinating thing about Lawsonibacter asaccharolyticus, in my opinion, is its main metabolic end products. This bacterium produces butyrate and acetate. So on the slide, I have the chemical structure of butyrate. And butyrate is a short chain fatty acid with several key roles in maintaining the structure, function, and health of the gastrointestinal tract. So, butyrate is a key modulator. Its role is so vital that disruptions of their abundance are often markers of certain chronic diseases. So, let's continue on. Let's talk about the article, what the author set to answer. Um, so, the researchers wanted to determine if coffee consumption consistently affects the gut microbiome composition across diverse populations. They wanted to identify specific microbial species associated with coffee intake. And in order to do this, they conducted a multi cohort, multi omic analysis of different populations with detailed dietary information. So let's move on. So the need. So coffee is one of the most widely consumed beverages, yet its impact on gut microbiome is poorly understood. So understanding how coffee interacts with the gut microbiome could help clarify the mechanisms behind these health benefits, offers potential pathways for dietary recommendations, and for microbiome targeted therapies. And so, the broader implication of the study, it advances the field of precision nutrition by demonstrating that a common dietary component can have a reproducible impact on the microbiome composition, and it could lead to personalized nutrition recommendations based on the microbiome profile of an individual. So I thought that was really cool. So I want to talk about a little bit about the cohorts that they had in their study design. So they conducted one of the largest metagenomic investigations of diet microbiome relationships. They used over 54,000 samples. So the image here is figure 1A in the article. And they had cohorts consisting of the Zoe Predict, and that was mostly individuals from the United States and the United Kingdom. They had what they called MBS, and that is the mind body study, and the MLVS, which is a lifestyle validation study. And so from those cohorts, it was a total of 22,867 participants, and they collected dietary assessments, stool samples, and blood samples from some of the cohorts, not all of them. And they also used 211 public metagenomic databases. So in red, we see some of the cohorts of healthy adults, children, and cohorts from non westernized populations, as well as non westernized populations of children. And you also see the NHP, and that cohort are non human primates. And they also were able to have access to ancient samples. So that's also indicated in the bottom there of the red public samples cohort. So they also got metagenomic data from uh, diseased adults, from westernized populations and non westernized populations, and from diseased children from westernized populations and non westernized populations. So this was a huge, huge study. So I just wanted to go over the cohorts a little bit. And so, this image, figure 1B, is the multi omic approach. And this, I just like the infographic to show that they conducted metagenomic samples, um, metatranscriptomics, and they, as you can see on the image, they only had 364 samples for the metatranscriptomics, and that was because that was the availability of high quality RNA that was needed. And they also had some blood samples, and those were from the MBS and the MLVS cohorts. And they also use the dietary assessments, the food frequency questionnaires, from those cohorts listed on the image, predict 1, 2, and 3 from the US, P3 from the UK, and from the MBS and the MLVS cohorts. And they applied some statistical analysis um, to categorize them as people who never drink coffee, which would be less than 3 cups per month, and moderate coffee drinkers, which mean 3 or less cups per day, or if they were high coffee drinkers, which meant 3 or more cups per day. And so I also wanted to dive in um, into all the methodologies that they used. So they conducted metagenomic sequencing and they used Illumina sequencing as the technology. And they were looking at the DNA extracted from the fecal samples. They conducted metatranscriptomics sequencing via Illumina sequencing. This is where they looked at the RNA from the fecal samples. They conducted metabo- m- metal- metabolomic profiling. And this was to see what metabolites were in the blood plasma. And they used liquid chromatography, mass spectrometry. And they conducted dietary assessments with what they called food frequency questionnaires. And that was just to uh, note the coffee intake between the cohorts. And so on my next slide, I wanted to talk about the bioinformatic tools and the statistical analysis that they used um, in their approach. So I have in pink all the statistical analysis that they used. So Spearman's correlation, it's a statistical method used to measure the strength and direction of a relationship between two variables. So they used this and they inputted the metagenomic data and the plasma metabolomic data, and they were trying to see if there was any correlation between coffee intake and microbial species abundance. And they used this algorithm called random forest classification. And that's a machine learning algorithm, and it makes predictions based on multiple decision trees. And so it allowed them to predict dietary patterns based on the microbiome composition. And they used receiver operating characteristic and ROC for short. This curve measures how well it classified people correctly. So it's a performance evaluation tool, and the value it gives, which is called AUC, area under the curve, um, indicates how good a model is at making the right call. So they used ROC on the random forest classification analysis to make sure it was making the right call by giving it assigning it to an AUC score. And so 1 meant a perfect classification, 0.8 or higher meant a strong predictive classification. Anything lower than a 0.5 was just as good as guessing randomly. They used a lot of bioinformatic tools as well. I just have 2 listed that I thought they were the most important. So I did a little bit of research on them. So Metaphi AN4 um, was used on the metagenomic data, and it's used for taxonomic profiling, and it's used to determine species level microbial composition. And the human 3.6 was used on the metatranscriptomic data, and that was for functional profiling, and it identified gene function in the microbe samples. And so let's continue on to our first figure. So figure 1C from the article, it really shows the predictive power of the microbiome. So graphed on the box plot is comparing two types of statistical evaluations related to how well the microbiome composition predicts coffee consumption. So on the y-axis, we have the Spearman correlation, and they put the metagenomic data and the metabolomic data into that. And we have the values on the y-axis from negative zero point 0.1 up to 0.6. And again, 0 means no correlation, 1 means a perfect positive correlation, and negative 1 means a perfect negative. And if that's a little confusing, so essentially, if a score was negative, it means the higher coffee intake meant the fewer microbes, where a 1, a perfect positive, would mean the higher coffee intake, meaning the more microbes. So that's what's being plotted on the y-axis. On the on the x-axis, we have the AUC value, and that was the evaluation of the algorithm, the random forest classification, and that was predicting coffee intake based on the microbiome composition. And so the values there are from zero point 0.5 up to zero point 0.9. And so anything that was zero point 0.8 or higher meant a strong predictive classification. And so the colored dots indicate the cohorts used. So they use from the Zoe Predict, Predict 1, Predict 2, Predict 3 UK, and Predict 3 US. And so the key takeaway from this figure is that among 150 food items, coffee has the strongest correlation with the gut microbiome. And so this figure, figure 2A, B, and C from the article, it's the Lawsonibacter drives association between the gut microbiome and coffee intake. So on the y-axis, it shows the top 10 species that show a strong association between their relative abundance and total coffee intake across 5 different cohorts. So on the bottom, we have our key that has the cohort symbols. And it's kind of divided into 3 from the total coffee, decaffeinated coffee, and prevalence. And on the bottom is the correlation number. And so the black markers that you see in the graphs indicate pre-cohort partial correlation. And the colored little boxes, the light blue for total coffee and the darker blue for decaffeinated coffee, this is the average correlation. And this is just an adjustment for confinders like sex, age, and BMI. So based on this graph, we can see that Lawsonibacter asaccharolyticus has a high prevalence, regardless of geographic or demographic differences. So it just shows the robust association with coffee consumption, regardless of the geographic or demographic differences. So figure 3A is showing the relative abundance of Lawsonibacter asaccharolyticus across different levels of coffee drinkers. So they had 3 categories of never drink coffee, moderate coffee drinkers, and high coffee drinkers. And they compared 1, 2, 3, 4, 5, yeah, 5 cohorts. On top, they have listed Predict 1, Predict 3 UK, Predict 2, Predict 3, and the MBS MLVS. And on our y-axis, we have the relative abundance in percentage. And on the bottom, we have the different categories of coffee drinkers. And so the key takeaway is that uh, the association across cohorts is not really significant between moderate and high coffee drinkers. Their prevalence of Lawsonibacter asaccharolyticus is basically the same, and those who did not drink coffee had lower abundance of that bacterium. Figure 3, figure 3B, figure 3B, C, B, C, D, and E pardon me, was the in vitro portion of this analysis. So they try to grow Lawsonibacter asaccharolyticus on an agar plate, and they did that again in a liquid medium. And they also try to grow E. coli and B. fragilis in a liquid medium. And they added coffee. And so they added on the bottom the color coordinated colors for each type of coffee they added. They had decaffeinated, instant, and instant decaffeinated coffee. And so for the liquid mediums on the y-axis, we have the OD, which is the optical density. And on the bottom, we have the coffee concentration. Whereas on the agar plate, we have CFU, which is just the plate count. So if we look at the two E. coli and B. fragilis, there was no stimulation. There was no growth even after adding coffee, decaf coffee. So that is not stimulated by coffee. However, the Lawsonibacter asaccharolyticus um, on an agar plate and on a liquid medium, adding coffee stimulated growth significantly over 350%. And that was regardless of caffeine. And this really demonstrates how coffee directly influences the microbiome composition. It actively stimulated Lawsonibacter asaccharolyticus. So on the next slide, I have figure 4A from the article, and they compare the prevalence of Lawsonibacter asaccharolyticus from 10 different host types. So on the bottom, on our x-axis, you would say, we have the different cohorts, and they kind of categorize them from healthy, from disease, to NHP, which is non-human primate and ancient samples. And they have the bottom, which has the cohorts, and if they use adults and from where. So non-westernized populations versus westernized populations, children versus adults, and they have them in group from healthy, disease, and diseased. So based on this figure, you can see that it's common to have a high prevalence of Lawsonibacter asaccharolyticus in Western populations, and it's rare in non-Western populations and almost non-seen, like, sorry, not seen in ancient samples. And it makes sense that there is a higher prevalence of Lawsonibacter asaccharolyticus in Western populations due to our modern dietary habits, particularly coffee. And it's probably more related to lifestyle than specific health conditions if we're comparing the healthy and versus diseased adults and children and their prevalence with Lawsonibacter asaccharolyticus. And I mean, Western populations, we have, higher coffee consumption, a lot of processed foods, and different hygiene practices. So I thought that was really cool that they were able to compare healthy to diseased people, but also Western and non-Westernized populations. And it was really cool that they had access to the non-human primate and ancient samples. So in figure 4B, this is showing Lawsonibacter asaccharolyticus prevalence and estimated coffee consumption across countries. Because they had access to open resources, databases, they had data from 43 different countries. And so here on this figure, they have 25, I believe. Yes, 25 countries. And on the bottom, that value is a kilogram of coffee per year consumed. And on the y-axis, we have the prevalence of Lawsonibacter asaccharolyticus. And so this indicates a strong positive correlation with countries with higher coffee consumption. They tend to have a higher prevalence in the bacterium, which makes sense. So for the discussion, I just wanted to restate my bluff. So what do we know now that we didn't know before. Coffee consumption is strongly associated with Lawsonibacter asaccharolyticus and independent of caffeine content. Lawsonibacter asaccharolyticus may play a role in mediating coffee's health effects through metabolic interactions. And what we know now, the increase is due to bioactive compounds, but not caffeine that stimulates the growth. And the broader implications is that diet can shape the microbiome in precise ways. And what we don't know are the long-term health effects of these changes. They remain unclear, so more study needs to be conducted. For the conclusion, coffee consumption is strongly reproducibly linked to the abundance of Lawsonibacter asaccharolyticus in the human gut microbiome. It highlights how a single dietary factor can shape the microbiome, influencing metabolic pathways and potentially impacting human health. And so I came up with 5 different questions to help us kickstart our discussion. So, what might be the health implications of increased Lawsonibacter asaccharolyticus levels? Could coffee be used therapeutically to modulate the microbiome? And what limitations do you see in this study's design? And how might different coffee preparations influence these results? People who drink coffee black versus, with cream or milk or with extra sweetness and 5 to 10 pumps of vanilla. And that concludes my presentation. Here on the last slide, I just have the references I used. So thank you so much for bearing that with me. Bearing with me. I hope you guys have a wonderful rest of your day.